For decades, Alzheimer's research has operated as a massive, high-stakes biological manhunt. Billions of dollars and countless clinical trials have chased individual proteins and receptors, yet the field is littered with late-stage failures. Take the most celebrated targets. Anti-amyloid antibodies successfully clear massive amounts of pathological plaque from the brain, but yield relatively minor cognitive improvements compared to the volume of clearance. Downstream neuroimmune interventions, like complement inhibitors designed to block the brain's immune system from eating its own synapses, have consistently failed to show durable clinical benefits. And attempts to directly stimulate specific microglial cleaning receptors, like TREM2 agonists, have fallen flat in advanced disease populations. These repeated disappointments point to a structural error in how we view the disease. We have treated the visible, surface-level pathologies, like the plaques, the inflamed immune cells, and the dying synapses, as separate, independent mechanisms to be targeted individually. The homeostatic matrix synaptic, or HMS, collapse model reframes this completely. It takes three historically divided pillars of Alzheimer's research and unites them into a single mechanical process. Sporadic Alzheimer's does not operate as a linear cascade triggered by a lone rogue pathogen. Instead, it is the simultaneous coupled failure of a three-layered defensive architecture that fails as a single unit. To understand how this architecture fails, we first have to look at exactly where it operates. The converging failures in Alzheimer's do not happen randomly across the brain's tissue. They center on a specific anatomical locus, the perisomatic zone of the parvalbumin-positive interneuron. This rapidly firing neuron represents the first layer of our system, the synaptic layer. Its primary job is to provide critical inhibitory signals that gate excitation across the cortex. Ensheathing this neuron is the second layer, the matrix. This is the perineuronal net, a dense geometric mesh of extracellular proteins that tightly wraps and structurally reinforces the neuron's main body. Because the PV-positive neuron fires so aggressively, it produces a heavy load of metabolic exhaust. The perineuronal net acts as a physical shield, catching and neutralizing these reactive oxygen species. Simultaneously, the lattice physically sequesters dormant pools of a vital signaling molecule called TGF-β. The third layer consists of the brain's resident immune cells, the parenchymal microglia, which patrol the outer perimeter of this protective net. As the matrix undergoes normal, healthy turnover, it continuously releases small amounts of that stored TGF-β, which binds directly to the patrolling microglia. This specific steady drip of TGF-β acts as a chemical leash. It maintains the microglia in a calm state and explicitly prevents them from reactivating aggressive developmental synapse pruning programs. The brain's cognitive health depends entirely on this continuous loop. The matrix protects the neuron and pacifies the microglia. In turn, the pacified microglia leave the matrix and the synapses perfectly intact. The onset of Alzheimer's pathology begins when cumulative, age-dependent oxidative stress disrupts this exact equilibrium. Over time, rising oxidative stress inside the patrolling microglial cells dysregulates a specific internal signaling cascade known as the SMAD pathway. As SMAD signaling stutters, the microglia lose their distinct healthy genetic profile, the Butovsky homeostatic signature, and warp into an inflamed post-homeostatic shape. Now hyperactive, these altered cells immediately secrete a wave of destructive effector enzymes, including matrix metalloproteinases and cathepsin S, directly into the extracellular space. These enzymes act as chemical shears, actively targeting and shredding the protective perineuronal net that surrounds the vulnerable PV-positive neuron. The degradation of the matrix triggers two immediate consequences. First, the loss of the physical net removes the local oxidative buffer. The highly active interneuron is suddenly exposed to the toxic force of its own metabolic exhaust. Second, and far more dangerous, the destruction of the matrix dissolves the localized reservoir of latent TGF-β. Without the continuous tonic release of TGF-β, the chemical brake holding the microglial population in check is entirely removed. The system is now trapped in a self-catalyzing cycle. By digesting the structural matrix, the immune cells destroy the precise chemical reservoir that was supposed to keep them from attacking the brain. This brings us to a specific molecular collision point, where the degraded matrix and the inflamed immune cells meet, the TREM2 receptor. As the perineuronal net breaks apart, the extracellular matrix fragments latch directly on to TREM2 receptors on the microglial surface. Instead of resolving damage, this binding acts as a molecular accelerant, amplifying the cell's phagocytic state and flooding the zone with more matrix-dissolving enzymes. This clarifies a long-standing paradox in the field. TREM2 mutations are a known Alzheimer's risk factor because the receptor attempts a tissue salvage program that, in an already compromised cell, produces catastrophic collateral damage. With the protective matrix gone and the TGF-β restraint completely depleted, the environment reaches a critical threshold. Dormant developmental immune programs, specifically the complement pathways, reactivate. These complement proteins drift into the perisomatic zone and chemically tag the newly bare, unprotected synaptic connections on the PV-positive neuron. Operating entirely without restraint, the hyperactive microglia recognize these tags, move in, and physically engulf the crucial synapses, stripping them from the neuron's body. The destruction of these perisomatic synapses causes critical inhibitory coverage across the cortex to fail, triggering a massive imbalance of excitation and a severe excitotoxic calcium influx in the cortical network. When this three-layered feedback loop becomes fully self-sustaining and destroys the brain's inhibitory circuitry, the system crosses an allostatic threshold. This is the mechanistic onset of clinical dementia. The ultimate test of the HMS collapse model lies in human tissue, specifically in the phenomenon of cognitive resilience documented in the 2024 De Vries cohort. This scatter plot comparing protein burden against cognitive decline highlights a distinct outlier group, patients whose brains possessed severe dementia threshold burdens of amyloid and tau, yet who exhibited zero clinical cognitive decline. If toxic protein buildup alone dictated the severity of Alzheimer's, these patients should have suffered from advanced dementia. The unifying biological characteristic of these resilient brains was the comprehensive preservation of the structural perineuronal net. In these outliers, the matrix remained completely intact. Furthermore, their microglia remained securely in a transcriptionally homeostatic state, and the synaptic contacts on their PV-positive interneurons were undamaged. The model predicts that protecting just a single piece of the system is futile. Having calm microglia but a degraded matrix guarantees an eventual propagating collapse. Cognitive resilience is not achieved by simply scrubbing away amyloid plaques. It is the specific biological state where all three layers of the homeostatic architecture are jointly and simultaneously preserved. This brings us back to the extensive clinical trial failures that have defined the last 20 years of Alzheimer's research. An anti-amyloid drug or a complement inhibitor fails because neutralizing a single downstream node does nothing to fix a collapsed upstream TGF-β signaling loop or repair a degraded matrix. True disease modification requires a strategy we can call homeostatic restoration. This means intervening pharmacologically to reinstate the upstream TGF-β and SMAD signaling pathways before the self-catalyzing loop takes over. Re-engaging this specific pathway simultaneously forces hyperactive microglia back into a surveillance state, restrains the complement pruning programs, and allows the perineural net to passively rebuild. The model points to a rigorous combination therapy. We need substrate reduction to lower the amyloid burden, combined with upstream signaling enhancement to restore TGF-β, and direct matrix stabilization to preserve the neuron's mechanical shield. Consequently, the ultimate measure of a drug's efficacy must shift. Verifying the physical integrity of the perineural net around these specific interneurons is a far more accurate readout of systemic brain health than measuring amyloid clearance. To finally halt Alzheimer's pathogenesis, the field must completely abandon the search for a singular linear pathogen. The ultimate solution lies in designing comprehensive interventions that fully respect and repair the brain's highly coupled, tri-layered organic network.